So guys, today I'm going to talk about glycolysis. Now, glycolysis is actually a part of cellular respiration, which is you and I breathing to produce energy. Now, glycolysis is what we call an anaerobic process. So anaerobic is the opposite of aerobic, which you probably know aerobic exercises is to get your muscles to ask for more oxygen. It's a breathing thing. Anaerobic does not require oxygen. So it doesn't require oxygen. And so the thing is, it needs some form of energy to get it pumping. So glycolysis, being an anaerobic process, it's a process to produce ATP, which I don't recall ever telling you guys what it is, but ATP is short for adenine triphosphate. And adenine is the same adenine that you find in phosphate is the same adenine that you find in DNA, which is usually called A. And its basic structure is that you have an A here, right, which is attached to a sugar, right? I'll just make a 6-carbon sugar. That's a horrible hexagon, but there's a 6-carbon sugar, which is then attached to 3 phosphates, which are represented by these P's. Now, a phosphate is PO4 negative. It's one of your polyatomic molecules that you learn about in chemistry. So anyway, to get back to the main topic, glycolysis, it's an anaerobic process that initially needs 2 ATPs. So you input 2 ATPs. And glycolysis is the phrase meaning breaking down of glucose. This glyco part stands for glucose, and lysis means breaking down. Okay? So if you go through this process in a bit more detail, it's a 10-step process, which I'm not going to get into all of the details of it, but basically what you need to know is you start off with glucose and you input 2 ATP molecules, and since it's an anaerobic process, you don't need oxygen, and it should give you 2 what we know as pyruvate molecules. I'll go into more detail about what pyruvate is, and it'll give you 4 ATPs and some ADPs and some phosphate groups, which is symbolized by Pi, which is inorganic phosphate. Okay, so as many of you already know, glucose is a 6-carbon molecule. Okay, it's a sugar, usually depicted as a hexagon with a bunch of double bonds, single bonds, whatever. And it usually has on each carbon an OH and an H attached to it to complete the 4 bonds for each carbon. So I'll just add them in. And so you have your glucose. Okay? So you have your glucose here. Now, the chemical equation of it is C6H12O6, right? Pyruvate, when you break it down, it's a 3-carbon molecule. It's usually linear. So you have a 3-carbon molecule, and you get 2 of these that form. I'm not going to add in the hydrogens and hydroxide groups, but it's basically this, and it's just breaking this down. So it's C3H6O3. And it's an energy molecule. Okay? This pyruvate moves on into different forms different processes depending on if there's oxygen present or not. So this pyruvate, if there's no oxygen, it'll go into fermentation, and depending on what kind of creature you are, if you're an animal like us, you'd be doing lactate fermentation, which will make lactic acid. And if you're a fungus or a bacteria, you'll be making alcohol or ethanol, and that's known as alcoholic fermentation. Now, if you have oxygen, then it will move on into the Krebs cycle, which will give you more ATP. Fermentation doesn't give you any ATP. It just brings you back to your original state. And that's what happens when you work your muscles a lot. When you're exercising, you don't get enough oxygen to your muscles, and so fermentation undergoes and you produce lactic acid, which gives you pain. So this is all I'm really going to get into for glycolysis for now. But just remember, you start with glucose, you input 2 ATPs, you get out 2 pyruvate molecules and 4 ATPs, giving you a net ATP count of 2. And this pyruvate can either go into fermentation if you're in an anaerobic, or goes into the Krebs cycle if you're in an aerobic environment. And next video you'll be seeing some gluconeogenesis, which is basically the opposite, but I'll tell you guys a bit of a twist. Subscribe to me so you guys can see some cool videos doing science and experiments and maybe learning something new. Thanks!